Hello, good afternoon, everyone.
Good afternoon.
Good afternoon, sir.
Hope you are doing well.
By
I'm doing well.
Yes.
And I
Thank God.
hand this up. Do you have any question from the previous lecture? All right. So I think we got to bacterial infections last week. Any confirmation? We did this, right? Did we look at this last week, please? All right. Loretta says no. What about this one?
No, sir.
We didn't get to this point.
Yes, please.
What about PCR steps?
No, sir.
Sir, viral life cycle.
All right. We did this.
Yes, sir.
Yes, sir.
All right. Thank you. So the viral life cycle, you know about the lysogenic and then the lytic. Yes, Josephine, your hand is up. Josephine. All right, let's move on. So the lysogenic has to do with the part of the life cycle of this virus where it does not kill the cell that it infects. It gets into the cell with its DNA or RNA. It incorporates or integrates its DNA into the host genome. So the host genome has part of the virus DNA. So anytime the cell replicates, the cell divides. If you divide with, please mute. Yeah, Joyce. Any problem? You can't see my screen. Any confirmation, please? Can you see this?
Yes, sir.
Yes,
Yes,
sir.
sir.
We can see.
Oh,
Yes, sir.
Manuela,
Yes,
how come
sir.
you
We
can't?
can see.
All right. So when the cell divides, it has the DNA on the virus that will also be divided and then multiplied. So it does not kill the whole cell. But we have the other life cycle, the lytic, where the virus gets into the cell. We introduce its DNA or RNA into the host cell, and then it will hijack the cell, destroy the host DNA. So you don't see the host DNA again. It takes over and then multiplies its DNA or RNA, and then gets assembled again with the capsid around it. And because the host cell DNA is destroyed, no activity can go on. So the cell eventually die. And then once the cell dies, the virus cannot survive. So it comes out to infect a new cell within the host. So the lytic has to do with the killing of the cell. through lysis. That's why it's called lytic, and then it comes out to infect new cells, and the cycle goes on. Sometimes they are strategic. When they infect another cell, they will integrate the DNA without killing the cell, so they can switch from the lytic to the lysogenic in the next phase. They are strategic. So the life cycle, we talked about attachment to the host cell, then they enter, then they replicate their DNA or RNA, then they assemble the coat, the capsid around it again, and then they come out. So that's for the lytic cycle. Then you can have some that do not destroy the cell. They will come out by forming vesicles around themselves, and then they are brought out or released for the next cycle to go on. So if it's about the lytic, and they always destroy the cell before they come out. For the lysogenic, they don't destroy, and they come out as burdens or budded entities, and then they get into the host to infect new cells. Viruses can be detected in so many ways. So we can monitor the immune response to the viruses. So immunologic assays that will look at antibody or cell-mediated activities. So once the virus gets into the host, the virus is an antigen, and the host will mount an immune response in the form of antibodies to attack the viruses. So anytime you want to measure the antibodies you have against, please mute, against some viruses, that means you should have already come into contact with these viruses before you can build antibodies. So usually if you don't have antibodies against these viruses, assuming there's an outbreak and this population is now yet to have the virus spreading to them, we can allow the viruses to spread so that naturally, yes, Elizabeth, your hand is up. We are just trying the options available anyway. So we can allow the viruses to spread. Mercy. Please, if you don't have any question or comment, put your hand down. So we can allow the viruses to spread so that naturally they build antibodies to know how the viruses look like, and then they can destroy them. Or you can present vaccines that have the antigens of the virus, the weakened form of the virus, so that your body will build antibodies in future to await the actual viruses that they will be encountering in subsequent days or years to come. You can also take specimen and then stain some tissues. Some of the viral infections will produce some pathologic effects. So you can use how the tissues appear to actually talk about or detect the presence of some of these viruses. So light or electron microscopy is possible once you stain these specific tissues or these specimens. You can also culture and then observe under the microscope to see these viruses. You can also use molecular techniques where you extract DNA or RNA of the virus, then you multiply, you amplify them, and then you can detect, because the numbers have now become many. So PCR, polymerase chain reaction, is commonly used for detection of viruses when we suspect such infections. And polymerase, that means it's an enzymatic activity that requires heating and cooling steps of the target DNA. And then the polymerase enzyme will add what you call nucleotides, the building blocks of DNA. And then you have several copies of the DNA or the target viral RNA or DNA amplified. Yes, Helen. All right. So for polymerase chain reaction, it's done in an instrument, an equipment. And this equipment is where you put your sample, which is your DNA or the RNA. So you take sample or specimen from the patient or the client, and if the person has the virus, you extract DNA or RNA of the virus, and you add components such as the primers. Primers are designed to bind to the target DNA or RNA. So they are specific. We have primers for HIV, we have primers for rabies. They are specific. Once they are able to bind to your target virus, then the polymerase can then add the nucleotides to build and extend this primer. You know, the primer is shorter than the target. So once the primer binds to the target, then the polymerase, which is the enzyme, will add the nucleotides to elongate so that you have a copy of the target doubled. So that is one cycle. Then the next cycle will begin where you have another binding. Then the polymerase will add the nucleotides so that you can multiply several times. All right. So PCR steps, polymerase chain reaction. It begins. Why are people raising their hands? Any problem, please?
Yes, sir. Please, I didn't get the polymerase chain reaction well.
Yeah, so let me use this to explain better. We have so polymerase chain reaction. It is an enzyme that is going to multiply or amplify the target viral or viral DNA or viral RNA. So it's about heating and cooling. When you heat, something happens to the sample. When you cool, something happens to the sample. So the DNA, when you heat, the double strands will separate to become single strands. And once they are single strands, primers can bind to each of the single strands. Then the polymerase will extend or add nucleotides to make the single strand now double strand because you have a primer which is specific to the target. So if I should use this to explain, you have your virus. Your virus is the DNA virus, double-stranded DNA. So once you heat the sample to about 94 degrees Celsius, this double-stranded DNA will separate. The hydrogen bonds between the two strands will separate so that you have single-stranded DNA because of the high temperature. Then remember I said it's repeated heating and cooling. So we have heated, the next is to cool. So you reduce the temperature to about 55 degrees Celsius. At this temperature, the DNA can come back to the double-stranded form. But because you have primers that are specific to the target DNA, they will bind. So here you are seeing a double strand, but the double strand is occurring at a very small portion, not the entire portion like we used to see here. Because of the primer annealing, primers will bind when the temperature is reduced to about 55 degrees Celsius. Please mute. We have feedback from you. Please mute. Then, once there is binding, it will occur or remain at this state until you increase the temperature again. At this time, not to 95 degrees Celsius or 94 or 98. If you increase to 98, it will become single strands again. But at 72 degrees Celsius, the polymerase enzyme will add nucleotides to elongate the primers, this one that's bound to the target of the virus, so that you have a double strand and another primer will also be extended or elongated. So you have two double strands. Initially was one double strand. So you have amplified this target virus to become double. So you have two of the viral targets. Initially was one. So this is one cycle of PCR. So it involves denaturation. This is denaturation. You heat the DNA, annealing, you cool the DNA to about 55 degrees Celsius, then extension about 70 degrees Celsius. So you end up doubling the initial viral target. Then you repeat the cycle about 35 or 40 times. So the next cycle, cycle 2, this one will double. So we end up getting 4 copies. Initially was 1. So now we are getting 4 copies. Cycle 3, it will become 8 copies because 2 exponent 3 is 8. Each of the 4 will now become double. So that's how PCR works. So your target is denatured, becoming single strands. Primers will bind to each target if you are specific. So if the sample is positive, your primers will bind. If the sample does not have the virus, there will be no denaturation, there will be no annealing, so there will be no extension, so there will be no doubling. That's how the sample will come out as negative when you detect. There's nothing to amplify, but if the sample has a virus, the primers will bind to them and then they'll be doubled. All right, so the one who has a question, Lisbeth, are you okay now? Yes, Ophelia.
Okay, so if I'm understanding you well, meaning that when the sample is taken from, let's say, an HIV patient who is having the HIV virus, and then the sample is being stained. No, if then the sample, when they add the primaries to the sample, then that is when the denaturalization will occur.
Yes, so let's take HIV for instance. You have a client, you want to screen, assuming the person came for A and C. You took blood specimen. Then because you want to extract and detect HIV. HIV is viral RNA. It's RNA virus. So you extract RNA. If you are not sure of the type of virus, we have a kit that will extract both DNA and RNA. Remember, if you are extracting DNA, human beings also have DNA. So you extract the virus DNA and then the human DNA in the same sample. But the distinction is you are targeting HIV for detection. So once you let me go back to this. So let's say this is the HIV, the virus that we have extracted is RNA virus. So RNA virus is single-stranded, mostly. In this case, the PCR, we do what we call reverse transcriptase PCR. You reverse transcribe. So transcription is you have DNA converted to RNA. That is transcription. Then the RNA is converted to proteins. Translation. This is how gene expression occurs in every organism. So for you to express any gene, maybe there's a gene responsible for your hair growth. That gene is in the DNA. That DNA will be transcribed into RNA. Then the RNA will then be decoded into proteins. So those proteins will then be expressed so that your hair can grow. The same way, because the RNA is in the virus, the HIV, we need to convert the RNA to DNA before we can start with the PCR. So that's why it's referred to as reverse transcriptase PCR. Once you get the HIV, the RNA of the HIV converted from RNA to DNA, then you can go ahead with the denaturation, annealing, and then extension. That is if the HIV is present in the sample. So if the HIV is present in the sample, primers will bind to the target DNA, and then the polymerase will add the nucleotides. But if the sample is negative, when you do the reverse transcription, because it only has the human RNA and then human DNA, your reverse transcription will not yield any results because you use specific primers for that. So the test will come out as negative. Is it clear, Ophelia?
Yes, please.
All right. So that's what PCR is about. You want to amplify a target DNA in the sample. The sample will have very small DNA. So you want to multiply it so that it can be detected. That's the purpose of PCR. All right. So after one cycle has denaturation where the DNA is opened up. Then annealing, you have primers binding, extension, the polymerase enzyme, adding the nucleotide so that you have double strand. So after one cycle, we have this one copy becoming two copies. Then after 2 cycles, this one will open up again. This one will open up again. So we have 4 single strands. Then the primers will bind to each of the 4 single strands and elongate it so that I have 4 double strands. So we have 4 copies after 2 cycles. So the next cycle, cycle 3, 2 raised to the power 3. So we end up with 8 copies because this one will open up, this one will open up, this one will open up. So all the 4 will open up. Then the primers will bind to all the 4. Polymerase will extend and then make all the 4 double. So if 4 is double, then it's 8. So that's how come the DNA becomes amplified at every cycle, copied, doubled, and then you end up having a lot. So after 30 cycles, 2 raised to the power 30, you can have about 1 billion copies of that virus made. And if initially you could not see those small viruses, the virus DNA, the virus RNA, because they were too small, because you have amplified and made several copies, now the machine can detect and say the sample is positive for HIV. Is it clear, please? Hello.
Hi.
Right. Thank you. So this is why we use PCR in most facilities. It's not only for viruses. You can use it for even TB detection. So the gene expert that they use, it relies on the same principle. You can also use place mute, another approach. So serological technique, where you want to measure immune response. So antibody or antigens in the fluids of the client or the patient. And once people get exposed to pathogens, they will build immunity. So we can do retrospective, go back into the population to see how many have been exposed to the coronavirus, how many have been exposed to the TB. Or you can measure the immune response to a vaccine, assuming we give vaccines to these populations. The vaccine is supposed to trigger immune response because it is a weakened form of the pathogen, so that the immune cells or immune system will produce antibodies, so that in case it meets the actual pathogen, it knows how to fight it. So if you want to know if the vaccine, is working, we measure the antibody levels. Then we screen for blood infections. We can use these serological techniques. So it works on antibody-antigen reaction. That's the principle. If antibodies are present in the person, once they are exposed to the virus, they will be binding, there will be an attack. The same way we can use this for testing using the rapid diagnostic method. If antibodies are not present, there will be no binding. So we can use an indicator, a control, to show us that the sample is positive or negative for the antibodies that we are going to test. So if the sample does not have the antigen, then the antibodies will have nothing to bind to. So the test will be negative. So you can have a clinical sample that has the virus, which is the antigen. The antibodies are produced within the host for immune attack. So on this cassette, you put your sample in a well. The sample has the antigen, and we have monoclonal antibodies that are pre-coated on the cassette. So nanoparticles on the cassette. And we also have a test line and a control line. These ones have anti-immunoglobulins that are specific for the target antigen. Then we have another control line that have antiglobulins that are non-specific. So the excess that are coming from this antigen will bind to the non-specific one, but this one, it will only bind to the specific antibody that is pre-coated here. So the samples will move by capillary action, and then it will sweep these monoclonal antibodies bound to the antigens, and then be presented to the antiglobulins that are specific for these antigens. So you see the binding here. Then we have the non-specific binding of these antibodies, monoclonal antibodies, which are already attached to the antigen. And then that's why you are seeing the yellow parts. This is non-specific binding. But for the test sample, the antigen or in the sample has to bind to the anti-immunoglobulins specifically presented by the monoclonal antibodies or test kit. This test kit is pre-coated with monoclonal antibodies that will bind to your antigen. So these are pre-coated on the cassette with good nanoparticles, and they are mobile. They can move by capillary reaction through the cassettes and then come to bind to will be presented to the specific anti-immunoglobulins for binding. That's why you are seeing the red here. These are the antigens from the sample. What you are seeing in yellow and the purple are what were on the cassette, the monoclonal antibodies. Then you have the control line, this one, also pre-coated with antiglobulins, but these are not specific to the antigen. They are specific to the monoclonal antibodies that were pre-coated here. That's why you are seeing them binding to your test line. That's why always the test line will show positive, because this one is monoclonal antibody. This one is the anti-immunoglobulin. So it binds to this one. So that's why the test line is always positive. But the control line, sorry, is always positive. But the test line has to bind to the antigen. The red has to bind to this one for us to see that it's positive. So this is how we interpret it. The control line is always positive, showing a band. And we look out for the test line. If there's a band, the test is positive because the test line is showing something. If the test line does not show anything, it is negative. If the control line does not show anything, don't come and look at the test line. It is invalid. We expect a band here before we come and look at what is here. So invalid could be control negative, the control line is negative, the test is positive, or both control and then the test lines are negative. When you see this, don't say it is negative because the control line is negative, it's invalid. All right. Millicent, are you okay?
Sir, please go back to the diagram and let me ask a question.
All right.
So, sir, when you put the sample on the sample pad, the antigen is on it already.
The antigen, if it is present, if you are, let's say you are targeting coronavirus, the virus is the antigen. The one that triggers the immune response is the antigen. What is inside? What is inside the host is the antibody. So the virus or the pathogen is the antigen. Is it clear?
So, sir, where from this?
Which one?
The antigen colloidal gold, the MABs,
Yes.
antigen colloidal gold. Is it the lab technician that will put it there, or is it the buffer that will be placed on the cassette?
Okay, so it's not a lab technician who is going to do this, or the lab technology. This is a kit. This is a cassette. The cassette comes with pre-coated. This is already pre-coated from the manufacturer. So they designed the kit for specific organisms. So let's say this is for coronavirus. So they have studied the virus to know the structure, and they'll design monoclonal antibodies that are able to bind to the antigen, the virus. If it's TB, they know how the TB looks like, the Mycobacterium tuberculosis. So the antigen, they have used that one to design monoclonal antibodies that are able to bind to the TB. So the antigen is the organism, the target that you are interested in, whether the virus or the bacterium or any pathogen. So these are already pre-coated on the cassette. So the sample, maybe sometimes the buffer will move the antigen for binding at this region. Then they will flow by capillary action. And then when it gets to the test line, the test line has anti-immunoglobulins. So take note, this is monoclonal antibody. So this binds to this one, presents the antigen, is specific. So it presents this antigen that is bound to the monoclonal antibody to the specific anti-immunoglobulin, this one. So if the sample is negative, it doesn't have the antigen, what happens is there will be no binding, there will be no presenting of the antigen from this colloidal section, the monoclonal antibody, to the anti-immunoglobulin. But because there's a control line, this control line is non-specific to the antigen, the one in red. So if you look here, you are seeing the binding of this anti-immunoglobulin to the monoclonal antibody here. Are you getting the difference? So that's why here you are seeing the binding of the red, that is the antigen, to the anti-immunoglobulin, the specific one. Here you are seeing the binding of the yellow. Yellow was not in the sample, it was already on the pre-coated section. So that's why the control line is always positive, because they'll be binding from this to this part. But for the test line to show a band, there must be a binding of the antigen to those anti-immunoglobulins. I get the difference now?
Yes, sir.
All right.
So, sir,
Hello.
for you to get a positive result, the virus needs to bind with the test line immunoglobulins.
Exactly, because the antigen is designed specifically to bind to these anti-immunoglobulins presented by the monoclonal antibody here.
Okay. So if the virus gets there and it doesn't bind to the immunoglobulins, but the yellow on the monocrobial antibodies, and it binds to the control line, it means it's negative or it's invalid.
Okay, so let me use this to explain. There must be a control band showing. So
Yes.
look at this one, the one in blue. Are you seeing the cursor?
Yes, sir.
Yes. These are anti-immunoglobulins. They are non-specific to an antigen. They bind to the monoclonal antibodies. They bind to these ones. And these ones are already on the cassette when the manufacturer produces them. And because these ones bind to the control line, even if you put water here, at the well, they will move this one to come and bind to the control line. So always you see a band. Are you following?
Okay. Yes, sir.
But this one will only show a band when the target is inside the sample. the antigen is inside the sample, it will not bind to any of the antigen in the sample before there will be a binding. That's how come always the control line is showing positive, but the test line only show positive when the target antigen is inside the sample. Is it clear?
Yes, sir.
Right.
Thank you.
Welcome. So you move on. So when you run the test, rapid test, rapid diagnostic test, and always the control line should always be positive. Always. You check the control line first before you come and check your test line. If there's nothing showing at the control line, it's invalid. So repeat the test. Use another cassette. Sometimes there could be errors, one in about 1,000. It depends on the cassette type. Sometimes if they are expired, you can be having a lot of errors. So if the control line is not showing any band, don't go ahead to look at the test line. It's invalid. Like infections through air, food, water, and other vectors. Environment that have these organisms that can cause infections. And the symptoms of bacterial infections are not specific to only bacteria. You can have viral infections or parasitic infections, and the symptoms also look like that of bacterial infections. That's why you need to diagnose properly by taking the right sample to test. And you have a lot of antibiotics available, so treating bacterial infections is not a problem these days. The problem is now that we have resistance. We treat, it doesn't go, we treat, it doesn't go, because we are abusing these antibiotics. And because we keep abusing, the bacteria will gain resistance over time because of drug pressure. And this affects morbidity and mortality rates in the communities. So when we say bacteria can cause infection, we look out for at least 3 features. In the community, if we get exposed to such bacteria, how likely will that bacteria gain access into the cells or the tissues or the organs, multiply, evade the immune system, and establish infection? That is the infectivity. So if you have 2 different bacteria, A and B, A gets into the system and begins to multiply, the immune system is able to control it, does not progress to infection. B gets into the system, multiplies, overcomes the immune system, and develops into infection. So B is infectious. So the infectivity rate of B is higher than that of the A. Then we come to pathogenicity. Pathogenicity has to do with moving from infection to disease. So that bacterium, its ability to cause disease after infecting the host is pathogenicity. Is it clear? Are you getting the difference between the infectivity and the pathogenicity?
No. No, sir.
Yeah.
Sir, please again. No, sir.
So this one is infectivity. Infectivity has to do with infection. Infection is about invading, colonizing, multiplying. So once they get access into the host, the immune system will try to attack, to destroy or kill. If the immune system is not successful, they will keep multiplying, invade more tissues or organs, and we say that because they are actively multiplying, there is an infection. So infectivity has to do with the ability of the bacterium or the pathogen or the organism to establish infection, multiply, invade in the living cells or tissues or organs. So in the community, if 100 people are susceptible and we expose these 100 people to the bacteria A, if bacteria A is not able to establish infection, but in the same 100 people, bacteria B is able to establish infection, the infectivity of bacteria B is of concern. That's why when there is an outbreak of two different organisms, we look out for the infectivity and then we prioritize one over the other. So pathogenicity has to do with the disease. So know the difference between infection and disease. Infectivity has to do with the infection. Pathogenicity has to do with after the infection is gotten or established, moving further to become disease. That is the pathogenicity. The ability of that bacterium or that pathogen or that organism to progress from infection to disease. That is pathogenicity. Is it clear now, please?
Yes, sir.
Then the third one
Yes,
is
sir.
about the disease. The disease, pathogenicity. But how severe is the disease? What is the measure of the severity, the pathogenicity? So that is the virulence. So yes, we can have these scenarios. Pathogen, bacterium A, B, C. A gets into the host and does not develop or establish an infection. So A is not infectious. When it comes to infectivity, it is 0. Then B is able to infect. Maybe the portal of entry is through the mouth. So you ingest contaminated food and then they attach to the intestinal walls and then establish themselves, multiply, they get the infection established. Then they begin to progress further and then cause the disease. So they are moved from infection to disease. So that B is pathogenic. A was not even able to establish infection and then move further to pathogenicity, cause the disease. And we have C, which is able to infect. So infectivity is achieved or the infection is established. It's able to move further to develop into a disease. And the disease is very severe. So the virulence of pathogen or the bacterium C is higher than that of the B. The severity that is associated with C is higher than that of the other organism. So we can compare the virulence Okay. hope it's clear now.
Sir, please, so
Hello.
what's the difference between the pathogenicity and the virulence?
Pathogenicity is the ability to cause the disease after the infection. So you should know the difference between infection and disease. You know the difference?
Yes, sir.
So that's pathogenicity. Moving from the infection stage to the disease is established. Infection is multiplying and then invading. Okay. Then we come to virulence, the degree of that disease, the severity. So the disease
Sir,
is,
please, can you say that the virulence, let's say the stage, in case maybe we are using cancer staging, can you
yes,
link the virulence maybe
yes.
stage 4 or stage 2 cancer or something?
So I don't want to use cancer because you can have cancer that is not as a result of an infection. I get my point.
Yes,
But
sir.
let's talk about malaria. We have severe malaria. We have uncomplicated malaria. So in terms of virulence, those that are having the severe form of malaria, that means the virulence of that parasite is higher compared to those that are experiencing the uncomplicated malaria.
Yes, sir, I
Or
get it.
let's say we have coronavirus infecting a population. Some will have the COVID-19 and they will just be having runny nose and coughing. But some will have the COVID-19 and they have to be admitted, serious condition. So the virulence of those COVID-19, the strains of the coronavirus is not the same. One is causing more severe form of the disease than the other. That is the virulence factor. Are you getting the difference now?
Yes, sir. Thank you.
All right. Jennifer, your hand is up.
Sir, please, I want to know what about gonorrhea? Is it an infection or a pathogenicity, a disease?
So gonorrhea.
Yes.
Gonorrhea is caused by Neisseria gonorrhoeae. So that's the bacterium. So the disease is what we refer to as the gonorrhea. But you can have Neisseria gonorrhoeae infection, which has not progressed to disease. I get the difference. So in terms of
Yes.
infectivity, we can say Neisseria gonorrhoeae infection.
Okay.
But once the disease is established, then this is what we are talking about as pathogenicity. That means the Neisseria gonorrhoeae is pathogenic because it has been able to progress from infection to disease. I get the difference.
Yes, thank you.
And some will develop very severe form. And the degree of severity, the measure of the severity is what we refer to as a virulence. Is it clear now?
Yes, sir, thank you.
All right. Yes, Beatrice. Beatrice and Grace, your hands are up. Oh, it's a mistake.
Hello.
Hello.
Please, so does it mean that if the body isn't able to fight against the infection, that is where the pathogenicity is established?
So sometimes, so let me start from this. The body may try to fight the infection, the organism. So the organism may get into the host, but will not develop into an infection. That is one. Infection is when the system is not able to contain them and they overgrow, they multiply. They become established in the site where they are needed. So that's when we refer to as an infection. So sometimes just exposure or incubation will not result in an infection. Are you getting the point now? But when the infection progresses, assuming it's not detected early, then they progress and then develop further, they become the disease. Is it clear now?
Yes, please. But so do we have a window period for every bacteria as to when it develops into a pathogenesis?
No, it depends on so many factors. One, it depends on the pathogenicity of that bacterium. It depends on the next slide we are going to talk about, the host. If the immune system is now able to mount an attack by producing more antibodies, so assuming the person takes the immune booster and is now able to mount an attack or start treatment early, or for those who usually have prophylaxis, they will be able to contain it and it may not develop into the disease state. So it depends on so many factors. Is it clear, please?
Yes, please. Thank you.
All right. Is that Beatrice or is a new person?
Yes, yeah, it's me. Sir, please, do we have like some bacterial infection that do not cause pathogenicity? As in the
Yes.
person will harbor the an example is Staphylococcus aureus.
Yes, the person could be
Like,
infected.
okay.
The person could be infected but not deceased, having the disease. But usually we see these people coming to the facility and then we term it as a disease because, of course, it will move from infection to the disease. But there's a thin line between infection and then disease.
Okay,
But
because I
generally, we
know
say the person
this.
has the disease. Yes.
There's this patient, like she did high vaginal swab and it came out as Staphylococcus aureus. Okay.
Mm-hmm.
And she treated it. She did another HVS and it came out as Staphylococcus non-aureus.
The coagulate is negative.
Coagulate, yes.
Mm-hmm.
Non-aureus. So, but she said she's still having like discharges from her vagina.
So sometimes, was she treated?
Yes. They
Mm-hmm.
gave her antibiotics. Yeah.
Sometimes you could have mixed infection. Mixed infection, and one of them will be leading to the disease. The other will be just infection. So if you treat, you may take care of the one that is causing the disease, depending on the antibiotic you introduced. So you eliminate the one that was having the ability to cause the disease. Then you have still the one that is causing the infection because the antibiotic you used was not able to fight or destroy that one. Probably is resistant to. So, and the coagulase negative could be normal flora also. So
Okay.
just detecting it in the sample does not mean it's an infection. To be a normal flora that is present at that site and not causing an infection.
Okay.
All right.
Okay. Thank you.
Right. But was the person also showing signs or symptoms? Sometimes the normal flora
she
would also behave.
doesn't get it.
Yes. So it could be normal flora, but in some cases, if that person's immune system becomes compromised, that normal flora could behave abnormally and result in an infection and disease. It depends on several factors.
Oh,
all right.
Okay, sir.
Yes,
Thank you.
You're welcome. We move on. Yes, so Grace is asking, what is a disease? Can someone tell her what is a disease? Yes, disease and infection. I think the first lecture I talked about this. Yes, who can try? Yes. Hey, midwifery, is it A or B? Midwifery A. Disease. Yes. A. Yes, Esther.
Okay, so disease is a condition that disrupts the normal function of the body or the mind, leading to signs and symptoms that affect health.
And infection. Infection.
We also said that the infection, it occurs when harmful microorganisms, that is the pathogens, enter the body and they multiply and causes damage to the tissue.
So when they cause damage to the tissues, would they result in disease?
Yes, yes, sir. Yes.
Yes, so
They result in disease.
So always, if it's about infectious disease, infectious disease will always be preceded by the infection. The infection comes first before the disease will occur. Those abnormalities that will be shown. Please mute. So usually, when there's an infection, because the pathogens, the organisms, they also have these properties to further cause disturbances and then disrupt the tissues and the organs. They progress to the pathogenic state, or they progress to become disease. Okay. So what you talked about earlier was about the bacteria, the one that is causing the infection and then disease. Now, you also have the host also playing a role. Sometimes the infectivity, how the organism is able to cause the infection, will depend on the host. So using the example someone just gave, the Staph aureus. The Staph aureus could be normal at that site, but if the host system changes, that normal could become abnormal this time. If there are underlying conditions, they could now change from normal flora to abnormal and become virulent. So the genetic makeup plays a role in the development of disease and infection. The nutritional status. If someone is malnourished, that person is at a higher risk of these infections developing faster into diseases. Age is also a factor. Children and those who are older, they also are at the risk of developing the disease from the infection. So somebody was asking about the window. It depends on these factors. Then the duration of exposure. Sometimes to the bacteria or the organism may be able to moderate within the host and then to survive longer. So the window between the infection and the disease is also elongated. Several factors come into play. And how long you get exposed to the same organism is also a factor. So if you are living with a carrier, always you're exposed to this carrier, the tendency that you develop the infection and then the disease is higher. So Salmonella typhi, the one that causes typhoid fever, if we have this carrier who is not aware of his condition, will keep spreading to people who are susceptible. So there will be infectivity by the Salmonella typhi that results in the infection. Then it progresses to the typhoid fever, the disease. Then you can have meningitis coming from those who are carriers of Neisseria meningitidis. They don't show any symptoms. They're just moving about. And then those who come into contact with them because of shared room or something, they get the infection. So Joyce is asking, can we say that the infection stage is the window period of the disease? The infection stage. Yes, so before, if it's about infectious disease, before the disease will actually manifest, or we say that there is a disease, that means the organism has been able to multiply. Sometimes they multiply, move from one side to the other, so that you can have infection from one side to the other. But once they get to the actual site, then the disease will result. Sometimes they will be at the same site, multiplying, and then the disease also results. All right, thank you. So, Dorcas, your hand is up. All right, let's move on. Dorcas, you can put it in the chat box. All right. You can also have these infections coming from animals. So zoonotic diseases gotten from infections from these animals that would further develop into disease. So somebody who lives with these animals or works with these animals is at a higher risk of a factor when it comes to the development of bacterial infections and diseases. You can have air pollution. So if you have people who are having the infection, the bacteria could come out of the infected people through the portals of exit, and whoever comes in contact with these sources will develop the infection if the immune system is not able to contain them. And some chemicals that we keep using or inhaling or encountering in the environment also weaken our immune system. And once the immune system is weakened, it makes way for bacterial infections. So hospitals where you have patients coming and going with different infections, this is a hotspot. So let's be careful when we are working in such environment. can have several sources of the bacteria agents that can cause infections. So waste disposal, they are channeled into the treatment plant, the liquid waste, then they get treated. The effluent is supposed to have no pathogen, but sometimes the plant could be malfunctioning, and then you can have the those who go to swim in there will also get the infection. Sometimes you have toilet facilities that are having the discharge directly into the environment, and then the infection could also be picked from there. Sometimes you can have these seeping through underground water bodies which are pumped to our homes, and the infection can also be gotten through that. You can have reservoirs where animals go and then drink, defecate, and can also have these animals being the source of the infectious agents. When they move around, then they also spread it. Can have transmission occurring in many ways. Can have direct contact, infected person, uninfected contact, and then there is transmission. Can have vectors coming to pick the infectious agent and then spreading to the other susceptible host. Can have food contaminated with dust that has a infectious agent. Can have flies settling on the feces or going to the refuse dumps to pick these infectious agents and then come to settle on the food. Can have water supply being another cause of these infections. People go fetch, then come and consume. Can also have droplet infection. So there are so many routes of transmission. Then clinical manifestations of these bacterial infections. I said earlier that the symptoms overlap. Sometimes you can associate some organisms or bacteria with certain conditions. So when you talk of meningitis, some of the bacteria that will come into mind is Neisseria meningitidis or Haemophilus influenzae. Hardly will you talk about skin infection that is associated with Neisseria, because that's not where they usually infect. But Staph aureus can infect so many areas. He's mute.
Remember. And that's the vision of this church.
Staph aureus can infect the skin, can infect the bloodstream, the abdomen, the lungs, so many sites within the body. So if in the sample that is analyzed, we report Staph aureus, we don't raise so much questions, but reporting Neisseria meningitidis when it's skin infection, that one is questionable. Some of these diseases can be as a result of distractions in the cells or the tissues, or the immune system trying to fight harder, and then there is imbalance. And this could sometimes lead to systemic inflammatory response syndrome as a result of large numbers of the cytokines or the other machinery that the immune system is trying to use to fight the infection. Or sometimes can have instability in the blood flow as a result of these bacterial infections that have progressed. And if treatment is not given early, most of them will develop into sepsis and sometimes results in the death of these clients or patients. So we can have several sites in the body or on the body that microorganisms or bacteria is mute. So when we find these bacteria or some fungi in these sites, it is no news because they are normal flora. Unless the immune system is compromised. So for some, like the HIV patients or clients, for those organisms in those sites, they could not be normal flora anymore because the immune system is now compromised. They cannot contain them as they used to be. Certain conditions or certain medications could also make these normal flora behave abnormally. Root. Oh, sorry. So we can have in the mouth, the nose, the throat, some of these bacteria, oral streptococci, Streptococcus pyogenes, Streptococcus pneumoniae, Neisseria species. So Neisseria can come or in the throat is no news because they are normal flora. But finding them in other sites could be a problem. So Staphylococcus epidermidis, Haemophilus influenzae, also in the throat. But once they get into the cerebrospinal fluid or the same for some of the yeast, these are not bacteria, these are fungi anyway. Then on the skin, we have a lot of normal flora. Staphylococcus epidermidis, Staph aureus, and have these Staph aureus in them. So they move from the skin this time into the lesions or the wounds and establish infections. Streptococci, Erythrois, Pseudomonas aeruginosa, all these can be harbored on the skin. But the problem is if you are collecting specimen, blood specimen from these sites and you don't clean well, you don't disinfect the sites, you may end up contaminating the blood sample or the specimen with these normal flora. Then you have perineum in the lower limbs having these fecal flora, the coliforms, and in the lungs, you don't expect any microorganism there. So any of these normal flora moving to the lungs will result in infection and subsequently disease. Then the stomach also is highly acidic, so not all organisms can live there, except those who are acid tolerant, like the lactobacilli, which you normally find in yogurt. Then in the small intestines, you have some microorganisms, same as the large intestines. So in stool sample, you can find a lot of microorganisms that are not reported as pathogens. The urethra and the vagina, you can also have some normal flora, the Staph epidermidis, diphtheroids, the streptococci, the coliforms, and then the anaerobes. But when these bacteria are found in these areas, then that is an infection because they are not needed in those sites. They are not harbored naturally in those sites. So you can have the central nervous system having these Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus influenzae, Streptococcus agalacea, Neisseria monocytogenes. And these will meningitis. We can also have viral meningitis. So taking the right sample to diagnose is also important. To have infections of the eye caused by Staph aureus, Neisseria. So Neisseria can cause meningitis and also cause eye infections. Chlamydia. You can also have the sinusitis, sinuses caused by Streptococcus pneumoniae, Haemophilus influenza. Upper respiratory tract infection caused by Streptococcus pyogenes, Haemophilus influenza. You have gastritis caused by several bacteria, several of them, depending on what you ingest that is contaminated. E. coli. Some of the E. coli are beneficial, but E. coli and then will also result in infection and disease. You can have urinary tract infections caused by E. coli, some of them. Then you have Enterobacteria, Staph aureus, Staph Staphylococcus, Pseudomonas aeruginosa. So remember, this was part of the normal flora, but this time it can cause UTI. Can have sexually transmitted diseases as a result of chlamydia, trachomatis infection, chlamydia, trachomatis infection, Neisseria gonorrhoeae, Treponema pallidum, ureaplasma, urealyticus, Haemophilus decree. Also have Staphylococcus aureus causing skin infections, although it could be normal flora. Can also have an infection caused by the Staph aureus. So you can have bacteria becoming normal flora, but ending up in other sites and causing infections. So fluconazole be taken for 10 days. The neighbor approached me with a so fluconazole. What's the condition? Usually, to administer antimicrobial agents, you have to know the condition and how that person is able to tolerate the antimicrobial agent. So please come again. What's the condition? So this is the end of bacterial infections. If there's no question, we move to the next part. Yeah, so Candida, this is fungal infection. So fungal infections, treating them, can have these Candida being normal flora. These Candida overgrowing and then resulting in the fungal infections, the condition. So this neighbor who approached you, was it confirmed Candida or it was a mixed infection? That's another question. If you have mixed infection, you may treat and then the other may not work. So some of the prescriptions depends on the condition. Depends on the condition. So we have standard treatment, but it will vary based on the condition that the person is presenting with. All right. It was only Candida species. All right. And also it depends on the strength of the fluconazole. All right. So we move to the next unit, specimen collection and processing, growth media and microbial culture, and then bacterial staining. So we start with the first part, general specimen collection and processing. So somebody is asking, what is the actual cause of H. pylori infection? So H. pylori infection, H. pylori is a bacterial infection. So it could be consumption of these infectious contaminated food or contaminated environment that the person comes in contact with. Usually, because it has to do with gastrointestinal, the person may be living with somebody who is infected and does not practice hygiene. But we have some carriers anyway. Some people are carriers, and such people could be able to transmit to others if hygiene is not practiced. So the portal of entry is mostly oral. All right. So if you want to properly diagnose, you need to collect the right specimen. So when you talk of sample is the part or the portion of the specimen that is measured, tested, or analyzed. So sometimes you use the entire specimen. So the entire specimen becomes a sample itself. Anyway, that's why we sometimes interchange these terms. So the right sample should be collected and transported in the right containers and should represent the condition. So if you suspect UTI, I hope you are not going to collect 2 samples, because urine will be representative of the disease or the infection. And when you are collecting samples for gastrointestinal tract infection, so the site should always be taken into consideration. If it's deep wound infection, you don't expect superficial specimen collection from these lesions. Otherwise, you may miss the correct cause of the infection. And timing is very important. If the sample has to be taken for bacterial culture, you advise that the person should have a sample or the specimen if the person is having severe malaria before they start the IV artesunate. Sample should be taken for malaria screening before the IV is initiated. Then the sample should be collected into the right containers, sterile containers. The technique should also be sterile, so that I don't produce any samples. The equipment being used should always be sterile. The right volume should be collected and they should be placed in the right containers. If it is blood sample, we don't expect blood sample to be collected in and then labeled. Containers being for sharp objects. Then the purpose of the specimen should also be indicated. That's why we have request Agnes. That's why we have requisition slip. So the results should also be communicated back to the healthcare provider. Communication. The patient name or the client's name should be on the requisition slip. The age, the gender, the patient ID or the hospital ID, the location where the sample was taken. Is it the ANC? Is it at the ward? These should be indicated. Sometimes a person could have multiple samples taken at different sites or locations, so they have to be indicated. Then the one who is providing care should have the name and the contact, if possible, for communication. The time of the sample or the specimen collection, the date as well, is very, very important. Especially when the sample is being transported, so that we can calculate how long the sample or the specimen has taken to get to that site. And if there is the request should also indicate the test needed. Oh, sorry, Rachel. So I'm saying that we have requisition slip. Is it better now, Rachel? Hello, Rachel. Please, can you hear me? All of you. All right, thank you. So each specimen needed for the test, you don't just take specimen for taking sake. There should be a purpose. So it comes usually on request, and it should indicate what is needed to be done, so that the information could be relayed back to the one who is requesting. But these days, because of the electronic system, you don't usually see samples or the specimen being transported with the requisition slip. Before the specimen gets to the lab, they have already entered everything electronically, and then the lab will know that this sample is coming. But the information going into the lab should always be accurate. Time is electronic. Hello? I didn't get you, please.
Yes, sir.
Just type of labs. Okay. Okay. So we have both ways. All right. Thank you. So this requisition slip or request form should contain the patient or the client's name, the age of the client or the patient, their gender, the hospital ID, the location where the sample was taken. So is it at the OPD, at the ward, at the emergency unit, or at the ANC? Or at the child welfare clinic? This should be indicated. Specimen collection, the type of specimen, very, very important. And the diagnosis needed. Sometimes one sample could be taken and then several tests will be required. So all these should be indicated on the form. Otherwise, the test which is needed, if it's not indicated, and this doesn't help the patient or the client anyway. And these should be transported as soon as possible. The earlier the better. And if it cannot be transported as soon as possible due to some delays, some specimen could be kept at tooth weight, so in the refrigeration temperature, or you can put them in the ice pack, the ice chest with the ice pack, and then the thermometer to monitor the temperature before they are transported. But for blood needed for culture, we have blood samples or specimen collected for several purposes. So if it's for culture, if it's not for hematology or for chemistry, but for culture, please do not refrigerate. Don't keep at tooth weight. If it's for culture and it's CSF, because there are some organisms, especially the fastidious or the anaerobic type, like the gonorrhea, the Bordetella, pertussis, the Trichomonas vaginalis. Please mute.
I'll beat you. Go.
These should be kept at room temperature. Room temperature is closer to the body temperature, 37. But if you keep them refrigerated, because this is very low, they may not adapt well, or it will take a long time for them to grow when they are brought into the lab. So we have exceptions for some of these samples. Always ask if you are not sure, so that the lab person could give you more information based on the test required. And usually for malaria screening, anticoagulated blood is needed and should be transported at room temperature. If for some reasons the site is far from the lab or the facility is far from the lab, the smear could be prepared by the lab person, and then you have the slide transported at room temperature. All right, so sometimes you hear or you see a samples or a specimen being rejected. The lab people don't take delight in just rejecting samples. We have criteria for assessing these samples or specimen before they are being processed. And please take note that rejection is not always outright rejection. Sometimes they are rejected, and if the correct information is provided, they are processed and worked on as soon as possible. Sometimes they could be rejected and recollection of the specimen will be needed. If improper transport temperature is used, it may compromise the quality of the sample or the specimen, so they may be rejected in most cases. Using the right transport medium container will make the sample or the specimen being accepted. But if it's improper transport medium or container, assuming you collect blood specimen and you still transport that blood specimen in syringe, mostly see this blood specimen collected in and kept transported in syringe, when the blood specimen could have been put into a right container or medium. So we don't encourage transporting blood in syringe with the needle also attached. Sometimes you see plaster being used to cover, or having the blood kept in the syringe and then wrapped with gloves. These are reasons for rejection. They should be put into the right containers. Prolonged transport time. If it is prolonged but not kept in the right transport condition, sometimes it could be prolonged but kept in the transport medium. So the time of sampling is very, very important, so that the person could calculate how long it has stayed between transportation and then collection. If the specimen is not labeled, who do we assign this sample to, or the patient to? So it could be rejected. If it's mislabeled, assuming you collected stool sample and then it's labeled urine, this can be corrected and then processed as soon as possible. But there should be communication anyway. If the container is broken, that one, it should be rejected. You don't know what contaminants have been introduced in there. If there is leakage from the specimen, it should be rejected. So these should be checked before they are even transported. Sometimes you receive samples in the lab and the ice chest, you open and there is leakage of the containers all over in the ice chest. And this can even be a source of infection to the ones who are going to handle the samples. Assuming it is airborne infection, the person could easily get it once they open the ice chest. Dried out specimen, these are not also accepted. That's why we encourage putting them in the right medium for transportation, especially if it's a swab specimen. Inappropriate specimen for the test requested. Malaria test and urine is collected. It's inappropriate specimen, so it should be rejected. Inadequate volume. In some cases, there could be an emergency, so inadequate volume could be accepted. But it should be taken into consideration the condition under which these inadequate specimens were collected. And if it's for culture, are going to grow the microorganisms, you don't keep them in fixative. Fixative will kill and preserve the cells. So we don't encourage keeping your specimen in fixative. Meanwhile, the organisms were there before they got into the lab. So it's bad to reject a provider why they were rejected. So mostly you see the lab people, they reject and keep with them without communicating. When you go back to ask for results, there should be communication all the time so that the patient or the client does not suffer. Incomplete information. This could be completed and then the sample or the specimen could be used again by the lab person. That's why I'm saying that sometimes rejection is not outright rejection. They could be accepted after the right thing is done. But it comes back to communication. Sometimes collecting the specimen again is difficult, especially if it had to do with surgical procedure and the procedure is over. Please mute. if the procedure is over, you cannot go back to the client and say you didn't collect the sample well, so you want to take her back to the theater. It's not acceptable. It's unethical. So the correct thing has to be done so that the sample could be used. If it had to do with mislabeling, the solution had to be provided so that the sample could be reused. So communication is very, very important. And whenever you are in the hospital or the clinic, always treat all specimens as infectious. Whoever client comes to you, you don't know the microorganisms they are harboring. So have at the back of your mind that you should protect yourself. And that precautions are very important. Wear gloves, your gowns or the lab coats, the nose mask, protective eyewear. In some cases, you need to wear them. And some facilities have areas that are designated for sample processing. So these have to be kept sterile all the time so that you don't have cross-contamination and causing infections as well. But we have specific recommendations for different specimen types. So, for example, blood, because you'll be taking the blood specimen from the veins, they're disinfected so that you don't introduce normal flora from the skin into the blood specimen to contaminate it. And if you are collecting from different clients, and it has to do with blood, I hope you are not going to use the same needle to collect these. Please mute. Please mute. When you join, you mute. And different containers should be also used when it has to do with different clients or patients, so that you don't end up mixing these specimens. If it has to do with blood culture bottles, sometimes a request will be made for aerobic and anaerobic bottles. So you should always label them using the right information from the patient or the client. So if 2 different bottles are needed for culture, aerobic and anaerobic, then different needles should also be used when you are introducing the specimen into these different bottles. And the timing is also important. If antimicrobial agents or antibiotics have to be administered, or they have to start anti-malaria treatment, the blood sample should be collected before the treatment is done. Sometimes you need to monitor treatment response. So after the treatment is initiated, sample or specimen collection will be required. That one is accepted because we already collected the first specimen before the administration of treatment. Somebody is asking the difference between aerobic and anaerobic bottles. So the blood culture bottles, if the request is for aerobic and anaerobic, the anaerobic bottle has oxygen depleted. So in that bottle, if there are microorganisms in the blood, organisms that are aerobic will not survive because oxygen is depleted. But the aerobic, you have some amount of oxygen dissolved and can support the growth of all obligate errors. You may also have some facultative anaerobes who can switch between aerobic and anaerobic conditions. Is it clear? Joyce? All right, let's move on. So in some cases, you may be requested to take different volumes depending on the test required. Remember, we are sticking to microbiology. Hematology, chemistry, analysis, you may be asked to take different volumes. So the volume you take for malaria test is not the same as the volume you take for blood culture. See, blood culture, you're taking a lot of blood. For malaria test, you'll be using just a small amount of blood. So the volumes are not the same, depends on the purpose. Please take note of that. Sometimes some facilities will request that you take this blood, and then when it gets to the lab, they will make aliquots and then use it for the specific test. So they can use malaria, some for malaria test, some for the blood culture, some for hematology. So it depends on the facility also. But if you are given the culture bottle, we have culture bottles for pedics, and that one usually you take 1 to 3 mils. We have culture bottles for adults, 8 to 10 mils. That one you are required to take between this range. But if it's for adults, we don't expect you to go and take the specimen from the adult into the pedic bottle. We usually see this happening. Or you take sample or specimen from the child into adult bottle. It's not encouraged. We have to use the right bottle if it's for children and then for adults. And keep them at room temperature. If it is for blood culture, take note, please. If it's for hematology, the condition will be different. But for blood culture, keep at room temperature. Don't keep refrigerated. For CSF, cerebrospinal fluid, should be transported as soon as possible. Sometimes there could be delays depending on the difference, the location of the two facilities. But if it's for culture, keep at room temperature. Assuming you want to test for meningitis outbreak, keep the sample at room temperature. But if it is for PCR, molecular test, where you want to extract DNA or RNA, you keep refrigerated. Please get the difference. So the purpose will always request or require the condition under which the specimen is stored. If it's for culture, you keep at room temperature, because remember, you're going to culture at 37 degrees Celsius. So room temperature is closer to the 37 than keeping at the fridge condition 2 to 8. All right. Sometimes, if it's for culture, you want to concentrate the microorganisms. So you may centrifuge, but this is mostly done by the lab people. Or you don't know where you find yourself in the future, a facility that will be requiring you to do that. In some cases, you may be ending up in a facility as a steady midwife or steady nurse for a project, and you may be required to do this. So that's why I'm adding this. So you'll be asked to centrifuge the CSF, and then you collect the concentrate, the sediment, which has the microorganism, and then it can be used for culturing. So for gastrointestinal tract infections, you can have fecal samples or rectal swabs in cases where the person is able to produce the stool or the feces. And usually for diarrhea or food poisoning detection. And stool is already having a lot of microorganisms, but use sterile container. Don't treat stool as stool. It should be put into sterile container and with a tight lid. Otherwise, there will be leakage, especially if it's watery stool. And stool specimen shouldn't be contaminated with urine. It happens mostly. Shouldn't be contaminated with urine or toilet paper. Some toilet paper will have these antimicrobial agents that can inhibit the growth of the specimen in the stool. And usually it should be transported at room temperature within 2 hours for examination. If it is for culture, it is advisable to keep them in the transport medium, like Stolz, Amis, or Caribbean medium. If it's for microscopy of intestinal parasites, we don't encourage keeping them in the transport medium because they're not going to culture. We're just going to examine by microscopy. It depends on the condition. Some cases they may request that you keep the stool sample in formalin for examination. So we always confirm before you start to add these formalin or put them in the transport medium. All right, so our time is up. We'll end here. If you have any question, you may ask, please. Any question, please? Yes, Georgina. Georgina, your hand is up. Yes, Georgina. Or is a mistake? Hello, yes. Please ask. Go ahead. Yes, Aisha.
Sir, please, does shaking of the sample affect the results? Let's say you want to know the HB level of a patient. Then you take the sample and sometimes you always shake it. So does that one affect the results?
Okay. This is microbiology, but let me answer that one. HB is for hemoglobin, right?
Yes.
Yes, hemoglobin. You have to keep the blood specimen in an anticoagulant. That's why we advise you gently mix it. And usually they use it for full blood count. Full blood count. So if it's just for HB, then that's when they use the HemoQ. But for full blood count, they put them in the FBC machine, and then it aspirates, and then you have the analysis or profile given. That's why we request that you mix it gently before some of the blood will get clotted. That's why you have the anticoagulant in the tube. But there is no microbiology anyway. Thank you. Yes, there was a hand up. Any other question, please? But for microbiology analysis, for infection, sometimes you need an idea of the HB levels. So, for instance, malaria goes with decline in HB. So some of them, as they request for the specimen for the microbiology analysis, they also request for the hematology as well. Any other question? Last question. Otherwise, we end here. Yes, Regina. Okay, so I think it was a mistake. All right. Enjoy the rest of your week. See you next week.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Right.
Or the idea